Welcome everyone. In this video, we'll discuss meiosis, the reduction division, and the ultimate source of genetic diversity. This is Meiosis Part 1. We will cover the introduction and detailed process of meiosis. Come, let's dive into the topic. Meiosis is a specialized nuclear division in which a diploid parent cell undergoes two successive divisions, Meiosis I and Meiosis II, but importantly, only one round of DNA replication. The result is 4 haploid daughter cells, each genetically unique. Remember, meiosis occurs only in germline cells, sperm, eggs, or spores, not in somatic cells. This is why meiosis is central to heredity and evolution. Formally, meiosis is a reductional division that converts a 2N cell into 4 haploid daughter cells via 2 sequential nuclear divisions following a single round of DNA replication. Key points to keep in mind: meiosis I is reductional, homologous chromosomes separate and chromosome number halves. Meiosis II is equational, sister chromatids separate, similar to mitosis, but in haploid cells. This two-step design creates both stability of chromosome number and genetic diversity. Here are the defining features in one place. Type of division: reductional, meiosis I, plus equational, meiosis II. Number of divisions: 2 successive nuclear divisions. DNA replication: a single round, premeiotic S phase only. Daughter cells: for haploid and genetically unique cells. Crossing over occurs in prophase I at the Pachytene stage. Genetic variation, achieved by recombination and independent assortment. Spindle formation, present in both divisions. Checkpoints: synaptonemal complex, SC, checkpoint, spindle assembly checkpoint, and G2/M controls. Why does meiosis matter biologically? It maintains chromosome number across generations by having gametes so that fertilization restores diploidy. It generates genetic diversity by 2 mechanisms: crossing over, exchange between non-sister chromatids, and independent assortment of homologs at metaphase I. This diversity is the raw material for evolution and adaptation. Meiosis underlies gametogenesis, spermatogenesis, oogenesis, and in plants, sporogenesis. Errors in meiosis cause aneuploidies, such as Down syndrome, trisomy 21, Turner syndrome, XO, and Klinefelter syndrome, XXY. Now, let's take a step back and look at the big picture of meiosis. This flowchart beautifully summarizes the entire process. At the very top, we start with meiosis itself, which is divided into 2 major stages: meiosis I, also called the heterotypic division, and meiosis II, also called the homotypic division. Comment what is heterotypic and homotypic divisions. In meiosis I, homologous chromosomes are separated. This stage includes prophase I, prometaphase I, metaphase I, anaphase I, and telophase I. But here's the interesting part. Prophase I is further subdivided into 5 substages: Leptotene, Zygotene, Pachytene, Diplotene, and Diakinesis. These are where the magic of recombination and crossing over happens. Then comes meiosis II, which is very similar to mitosis. It includes prophase II, metaphase II, anaphase II, and telophase II. Here, the sister chromatids are finally separated, leading to the production of 4 genetically unique haploid gametes. So, this slide is your roadmap of meiosis. Meiosis I reduces the chromosome number, and meiosis II ensures each gamete gets just 1 copy of every chromosome. Together, they guarantee both genetic stability and diversity. Premeiotic interphase. Before any meiotic event, the cell completes interphase G1, S, and G2. In G1, the cell grows and prepares biosynthetically. During S phase, DNA replicates, so each chromosome now consists of 2 sister chromatids joined at the centromere. G2 is for assembling proteins required for meiotic division and for DNA repair. Unique to meiosis, meiotic-specific proteins such as REC8, which is a meiotic cohesin, and SPO11, which initiates recombination by creating double-strand breaks, are expressed. These replace some mitotic factors and set up recombination and special cohesion patterns needed for meiosis. Now let's look at the Meiosis I, which is a reductional division, where the homologous chromosomes separate, reducing chromosome number by half. It is called heterotypic division because the two daughter cells produced are genetically different from the parent cell and from each other, primarily due to the separation of homologous chromosomes, the process of crossing over, and the subsequent reduction in chromosome number from diploid to haploid. Now, let's begin with prophase I. Prophase I is the longest and most complex phase and is subdivided into 5 substages. Use this mnemonic to remember them. Little zebras play during daylight. Leptotene, Zygotene, Pachytene, Diplotene, Diakinesis. I'll walk you through each substage now. In Leptotene, chromosomes begin to condense and appear as thin threads. Sister chromatids are already held together by cohesins. A notable event is the bouquet arrangement where telomeres cluster at the nuclear envelope. This nuclear organization facilitates homologous pairing. In the next subphase, Zygotene, homologous chromosomes start synapsis. They pair tightly. The synaptonemal complex, a proteinaceous zipper-like scaffold, begins to form between homologs. Proteins such as SYCP1, SYCP2, and SYCP3 are components of this complex and are essential for stable pairing. Next comes Pachytene, which is the critical recombination stage. Here, crossing over actually occurs. Double-strand breaks are introduced by SPO11, then recombinases, RAD51 and DMC1, mediate strand invasion and exchange between non-sister chromatids. These exchange events become chiasmata, and they create new allele combinations. This is the molecular basis of genetic recombination. In the next subphase, Diplotene, the synaptonemal complex dissolves, and homologs begin to separate but remain connected at chiasmata, the cytological manifestation of crossovers. Chromosomes are highly condensed in this stage. Importantly, in many animals, oocytes arrest at diplotene, dictyate, until ovulation, an important physiological feature.
Diakinesis is the terminal stage of prophase I. Chiasmata terminalize toward chromosome ends, chromosomes become maximally condensed, nuclear envelope breaks down, and spindle microtubules attach. Checkpoint note: the Pachytene checkpoint ensures recombination is complete and correct. If recombination defects are detected, the cell can arrest or undergo apoptosis, a safeguard against defective gametes.
In metaphase I, the bivalents, paired homologous chromosomes, align at the metaphase plate. The orientation of each bivalent is random with respect to maternal or paternal origin. This randomness is independent assortment, a major source of variation. For humans, independent assortment alone yields 2 to the power of 23 combinations, that is over 8 million. During anaphase I, homologous chromosomes separate and are pulled to opposite poles. Notice that sister chromatids remain attached at their centromeres. This sister cohesion at centromeres is protected by Shugashin, which prevents premature separation, a crucial distinction from mitotic segregation. Telophase I. This is Th last phase or meiosis I. Now the nuclear envelopes may reform depending on species, and cytokinesis divides the cytoplasm, producing 2 haploid daughter cells. Each daughter has 1 set of homologs, but the chromosomes are still duplicated. Sister chromatids are present and will separate in meiosis II. Interkinesis. Between meiosis I and II there may be a short rest called interkinesis. Importantly, no DNA replication occurs during interkinesis. The cell prepares for the second division, synthesizing proteins and reorganizing the spindle. Now let's look at meiosis II. Meiosis II is the equational division. It resembles mitosis, but starts from haploid cells. The key outcome is separation of sister chromatids. The phases are prophase II, metaphase II, anaphase II, and telophase II. In prophase II, chromosomes recondense if they decondensed. the nuclear envelope breaks down where present, and spindle fibers form in each haploid cell. This sets up alignment for metaphase II. In metaphase II, chromosomes align individually at the metaphase plate, and each sister chromatid's kinetochore attaches to microtubules from opposite poles. This is like mitosis, but with half the chromosome number. Anaphase II sees cleavage of centromeric cohesin by separase, allowing sister chromatids to separate and move to opposite poles. This completes the physical separation of the duplicated DNA into distinct chromatids. Finally, in telophase II, chromosomes decondense, nuclear envelopes reform, and cytokinesis yields 4 genetically unique haploid gametes. Note, in spermatogenesis you get 4 functional sperm, whereas in oogenesis cytokinesis is asymmetric, producing 1 large ovum and polar bodies. A quick comparison table, memorize it. Division rounds, Mitosis I, Meiosis II. DNA replication, both do 1 S phase preceding division. Homolog pairing, absent in mitosis, present in meiosis, synapsis. Crossing over, absent in mitosis, present in meiosis. Outcome, mitosis gives 2 diploid identical cells, meiosis gives 4 haploid genetically varied cells. This table is frequently tested in Part B questions. And that's a wrap for Meiosis Part 1. We've covered the introduction and complete process of meiosis, how 1 cell becomes 4, each genetically unique. In Part 2, we'll explore microscopic observations, experimental proofs, and the clinical importance of meiosis. Don't forget to like, subscribe, and share this video, and check out the quiz shorts in the description to test yourself. See you soon in Part 2 on Sravika's biology.